I am
David Wong at the University of California, Los Angeles. We're very privileged to be advancing from ERCC Stage 1, which our project is to translationally and scientifically underpin how biomarkers could traverse from an organopathology, in our case here, gastric cancer, and how these organ-specific extracellular RNA could traverse itself from the organ pathology through vasculature to the salivary gland into saliva. So stage 1 is a translational validation, a definitive translational validation of biomarker development using extracellular RNA in saliva. And stage 2, as it evolved to be, we have that all-important mechanistic opportunity to underpin why is systemic disease that's at a distal locale to the salivary gland could convey the information of a diagnostic potential through this vesicular transport through the vasculature and come into the salivary gland and into saliva for us to design a host of translational and clinical utilities. How does the research in stage 2 fit into my research lab? It really dovetail and forward in a wonderfully important manner, in the sense that saliva liquid biopsy is our main research dimension, namely the utilization of this biofluid that each one of us produce 3 soda cans a day, that we can harness non-invasively, non-painfully, and non-embarrassingly, that it contains these omics constituents that could allow, personalized medicine, precision medicine type applications. And in this scenario here of ERCC2, how and why a systemic cancer, gastric cancer, could convey its pathology-specific biomarker, extracellular RNA, through vesicular transport, come into saliva, that could be utilized for clinical early detection of the disease, monitoring of treatments, amongst other clinical utilities. So it really fits into, in a very good way, where the frontier of liquid biopsy and how saliva could be that clinical sample type that could be engaged in this frontier. It's a very important question. When did I first realize of the existence of extracellular RNA, and perhaps in the context of the community and the field? Serendipity 12 years ago, when we independently discovered extracellular RNA in saliva as a biomarker for oral cancer. Makes sense. It's a local malaria to oral cancer. And in addition to proteomic targets, we found a panel of extracellular RNA that exists in the saliva of head and neck cancer patients. We published that. It gets quite a bit of visibility. NIH actually has a press release on it, but it was met with tremendous skepticism, and rightfully so, because how could the labile molecule RNA exist in a hostile environment? Saliva is often viewed as a hostile environment because it's exogenous, communicated with the exogenous environment and the existence of microbial targets. So it was challenged, if that's a good word for it, until the ERCC1 came along 7 years ago and overlay this transformative domain where extracellular RNA is being valued, nurtured, and developed in a biomarker perspective, therapeutic, basic science, and profiling. It's a privilege to be in ERCC2, and I'm sure every membership feels that way. Nurture it, embrace it, and advance the science. And it's truly transformative, it's paradigm changing, and it's a privilege to be in ERCC2.